Buffers. Okay, so if we're going to have a stable pH despite adding and taking away a hydrogen ion, okay, then we're going to need to have buffer systems. We have different buffers inside of cells than outside of cells. Our primary extracellular buffer is the one I just showed you. This is the carbonic acid buffer system, right? The bicarb carbonic acid buffer system. And it's that equilibrium. Now, a thing you need to know, in the body, H2CO3 doesn't happen for very long, okay? So like, you're never gonna have much carbonic acid. We have an enzyme called carbonic anhydrase that will either break it into water and CO2, that's carbonic anhydrase, or carbonic acid at our body's temperature and pressure is going to dissociate into bicarbonate hydrogen ion. All right, so this is an intermediary. The only reason I put it in there is I want you to see that all the atoms are the same, right? We're not adding or taking anything away. We're just changing the form. Okay, so since this is our primary extracellular buffer, it means that the lungs can effectively get rid of a hydrogen ion by blowing off a CO2, right? So if we take a H plus and a bicarb and make carbonic acid falls apart into H2O and CO2, we get rid of the CO2. So what have we done? We got a water in exchange for a bicarb, and we used up a hydrogen ion, right? So we effectively elevated our pH a tiny, tiny little bit, right? So this system of breathing faster to increase the pH, breathing slower to decrease the pH, is our primary minute-to-minute acid-base balancer. To such an extent, as we'll learn in the respiratory regulation stuff after the break, that the body actually controls the rate of respiration and depth of respiration, based on pH more than anything. All right, so, and we'll see that over and over again. The kidneys, however, do still play an important role. What the kidneys are doing is they're dealing with all of the other kinds of acids that the body can produce, that this system, the respiratory system, can't deal with. For the lungs, if it's not water, CO2, or bicarb, it can't do anything about it. So the kidney they're much slower at adapting to acid-base challenges, but they can work on all the acids, not just the volatile acids. A volatile acid is one that has a component that bubbles out into the air, right? So carbonic acid, when you crack the soda pop, right, you get bubbles. Why? Because the CO2 came out of solution. And while the chemistry is maybe a little different in the body, the net result is still the same, right? That if we breathe out, we've taken away a hydrogen ion. Okay, so what we see over here is the pK curve, the buffer curve of carbonic acid or CO2 versus bicarb versus pH, right? So here is amount of CO2, it's inverted, okay? So high CO2, low pH, right? There's a relationship that will always be true. Down on this side, high pH, low CO2, all right? Now, we can also see that at high pHs, most of our equilibrium reaction is living here, 100% of the carbons are living as bicarb, right? Whereas down here, 100% of the carbons are living as CO2 or carbonic acid, all right? So we see this range and the nice pretty buffer curve. Now what's interesting is our buffer system has a pK that's like 6, that would be at balance 50-50 at like 6.1. That isn't where the body lives. We live way up here. So even though this is our buffer curve, the body prefers to have less CO2 and more bicarbonate, right? So our system lives up here instead of down here on the steep curve. We live up here on the flat part of the carbonic acid bicarbonate buffer curve. All right, so again, you don't have to calculate anything, but I do want you to know that we're not at the equilibrium point, right? We're actually way up here towards the bicarbonate side. Okay, so extracellular fluid. That's where this plays into its role, okay? The carbonic acid buffer system is an extracellular fluid plasma blood buffer system. Inside of cells and in other bodily fluids, the buffer systems are a little different. Okay, so inside of cells, phosphate plays a major role as a buffer. And if you'll remember from your chemistry, phosphate has more elaborate buffer systems, right? Because there's more than one, valence state or whatever they call it for that one. Again, I just want you to know phosphate is buffer in ICF. Right? Don't worry about the equations right now. Yeah. Oh, okay. So that's one, phosphate. Phosphate's all around ATP, right? And the nucleotides, right, have a phosphate component. Proteins. Inside of cells, lots and lots and lots of proteins. Proteins tend to have regions that are negatively charged. Negatively charged regions hold on to hydrogen ions. That makes proteins a buffer. Right? Because they can hold on to hydrogen ions and they can release them depending on what the pH is. So those are two primary intracellular buffers. Most of the acid base, because of our ICF volume is quite a bit higher, and because intracellularly is where the metabolism is happening, 60% of our buffering is happening intracellularly. All right. Now, hydrogen ions and bicarbonate, believe it or not, are not particularly permeable. They need a transporter. So you're going to have some cells are going to be more permeable to bicarb and hydrogen ions than others. So what that means is that the hydrogen ions are exchanged. In other words, the extracellular fluid and intracellular fluid exchange hydrogen ions and will have an equivalent pH. But it will take time, right? So the intracellular buffers can take many hours to functionally lower the ECF's pH or raise it. So ICF and ECF both have pH. They're usually the same. If one compartment changes, it will take some time, hours, for the other compartment to change to match it. Okay, which means that we can't just give, a whole bunch of bicarbonate and let the pH just magically get better, right? Because you have intracellular and extracellular effects. So it's